Hello, I am Agnieszka Cecotka, and I would like to present my work entitled DNA methylation alterations in de novo and therapy-related AML in several genomic regions. Acute myeloid leukemia is the malignancy of myeloid stem cells, which don't develop properly into myeloblasts and then into monocytes and granulocytes. Instead of this, immature abnormal cells are produced and accumulated, but the production of normal blood cells is impaired. Why does it happen? From all of the possible reasons, I would like to mention two. The first is AML development by itself. The reason is unknown and we call it de novo AML. The second is the side effect of radio or chemotherapy of previous cancer given several years before. We call it therapy-related or TAML. Patients with TAML characterized with the worst response to treatment and the worst prognosis. The reason of these differences can be epigenetic alterations. One of the epigenetic processes is DNA methylation, which controls gene transcription. It's a change of cytosine into 5-methylcytosine in the CpG sites of the genome. CpG site is cytosine followed by guanine in the DNA sequence. DNA methylation has the most impact on gene expression when it occurs in CpG-rich gene promoters. If promoter methylation level is low, transcription factors can bind to the promoter and the gene is expressed. When the promoter is highly methylated, transcription factor binding is blocked and the gene can't be expressed. In cancer diseases, the first situation applies to proto-oncogenes whose expression is increased, and the second is for tumor suppressor genes whose expression is stopped. The data which I have analyzed were obtained with Illumina Infinium human methylation epic array, which allows for measurement of methylation level across the whole genome in over 850,000 CpG sites. CpG sites are annotated to the areas which construct genomic regions, CpG-rich regulatory sequence regions, gene body regions, and free pre-mutual regions. The number of CpG sites annotated to each type of region is presented in the circle diagram. The data was obtained for 3 patients groups: healthy control 5 people, de novo AML 5 patients, and TIML 6 patients. Infinium human methylation epic array can measure methylation level from only 8 samples, so for 16 patients, 2 arrays were used. This can cause the batch effect, which may result in the differences between these two sample groups. So the first step of the analysis was batch effect removal. In the graphs, 2 first principal components are shown before and after this procedure. After batch effect removal, samples for both AMLs are more coherent. Methylation level is measured as beta value between 0 and 1, where 0 means no methylation and 1 means full methylation. I looked at methylation level distribution in each sample, and I presented it on the empirical CDF graphs. Distributions are diversified among patients, even within the same patient group. Then I looked at methylation level distribution for all types of genomic regions. CpG-rich regulatory sequence regions differ most from other types of regions. I compared methylation level in each CpG site among all patients groups with an ANOVA test. And the results are presented in the figure. 33% of all CpG sites are differentially methylated in at least one of the patients groups. When we look at genomic regions, 35.5% of CpG sites are differentially methylated in CpG-rich genomic regions, almost 34% in gene body regions, and 32% in free pre-mutual regions. I would like to focus on CpG-rich regulatory sequence regions because their methylation has the most impact on gene expression. Out of these 35.5% sites, when we compare de novo AML with control, over half is upmethylated and 4% are downmethylated. And when we compare TIML with control, almost 90% of sites are upmethylated and 6% are downmethylated. It's a big difference. So let's compare TIML and de novo AML. Out of 33% sites differentially methylated in the whole genome, the amount of up and downmethylated sites is 14% each. In CpG regulatory sequence regions, 30% were upmethylated and 3% were downmethylated. In gene body regions, 12% were upmethylated and 13 were downmethylated. In free pre-mutator regions, 11% were upmethylated and 12% were downmethylated. To summarize, empirical CDF graphs show diversification of methylation value distribution among patients, as well as differences in distribution among genomic regions. Upmethylation is a process stronger than downmethylation in the novo AML as well as in TML in comparison to control in every type of genomic regions. The differences between AMLs and control are stronger than between de novo and TAML. The biggest contrast between AMLs is observed in CpG-rich regulatory sequence regions, which have the most significant impact on gene expression. I would like to thank the authors of this proceeding and thank you for your attention.